Nobel Laureates, representatives of Thailand networks, administrators of academic institutions, chief executive officers of private sectors, honorable guests, dear participants, ladies and gentlemen, welcome to the 5th ASEAN event series on Bridges Dialogue Towards a Culture of Peace, hosted by Bridges Dialogue Towards a Culture of Peace, International Peace Foundation and Naresuan University, co-organized by the Thailand Network. I am Pongam Liamsiriwattana, a lecturer from Naresuan University International College, and the lady standing next to me is Dr. Chayanin Pratumsood, a lecturer from the Faculty of Public Health, Naresuan University. Today, we both are truly honored to serve as the MCs of this great event.
Thank you very much for your kind introduction. This year, ladies and gentlemen, our event is kindly honored by Professor Bruce Alan Beutler, a 2011 Nobel Laureate in Medicine, who is also the director of the Center for the Genetics and Host Defense at the University of Texas Southwestern Medical Center, Dallas, USA.
Well, Dr. Chayanin, let's talk about the special lecture today, the global struggle against infectious disease. Personally, I feel that the issue of transmitted disease on a global level is something that we should be aware of.
Yes.
And it's been a decade since we experienced the outbreak of anthrax, bird flu, and recently, Ebola.
Yes.
So today, we are honored to have Professor Beutler with us, whose lecture is directly related to the current health issues that I just mentioned. So as a person who has been working in the medical science and public health, could you please give a brief introduction about Professor Beutler and his work, please?
Yes, of course. His tremendous achievement was the discovery of an important family of receptors that allows mammals to sense infections when they occur, triggering a very powerful inflammatory response. Professor Beutler is known for his pioneering molecular and genetic studies of inflammation and innate immunity. He was the first to isolate the mouse tumor necrosis factor alpha, or TNF-alpha, to demonstrate the inflammatory potential of this cytokine, proving its important role in endotoxin-induced shock. Subsequently, he invented recombinant molecules expressly designed to neutralize TNF, fusing the binding portion of TNF receptor proteins to the heavy chain of the immunoglobulin molecule to force the receptor dimerization. And these molecules were later used extensively as the drug etanercept in the treatment of rheumatoid arthritis, Crohn's disease, psoriasis, and other forms of inflammation. Professor Beutler has been elected to numerous honorary academic societies. These include the United States National Academy of Science and the Institute of Medicine. Professor Beutler is also an associate member of the European Molecular Biology Organization, or EMBO, and a member of the Association of American Physicians and the American Society for Clinical Investigation.
That's very interesting. And I myself, as a person who's working in social science field, I also think about global health issues or global health security. I mean, in the 21st century, health is a shared responsibility involving equitable access to essential care and corrective defense against transnational threats. So I believe that we and our participants will enjoy the special lecture from Professor Beutler in the next coming sections. By the way, for now, let's get back to the conferment ceremony. At this event, Naresuan University takes this opportunity to confer an honorary doctorate in medicine on Professor Bruce Alan Beutler, who is recognized as the world-renowned figure. In addition, there are some main sponsors that should be mentioned here. First, Cognita Asia Pacific, second, Central Hotels and Resorts, and third, Millennium Hilton.
So thank you once again for your kind support. This year, ladies and gentlemen, we are celebrating the 21st anniversary of the founding of the university. Prior to the conferment, before we are going to hear such a remarkable speech from Professor Beutler, we would like to show you a short video presentation of Naresuan University, which takes place at the Pitsanulok province, where the university is situated.
So if you are ready, let's see the presentation, please.
It has been 21 years since the official inception of Naresuan University, named after King Naresuan the Great, on July 29, 1990. Dedicated to the mission that Thai society be free from ignorance, the university has pledged itself to effective knowledge contribution to produce students who will become good citizens of the country. This will not only assure a better quality of life in the society, but also the continued development of human resources in Thailand. Presently, the university accommodates 5 colleges or schools and 16 faculties. The 5 colleges or schools are the Graduate School, the School of Renewable Energy Technology, Naresuan University International College, the Logistics and Supply Chain College, and the Institute for Fundamental Study. The 16 faculties can be grouped into 3 clusters. The Health Sciences cluster comprises 7 faculties: the Faculty of Medicine, the Faculty of Pharmaceutical Sciences, the Faculty of Dentistry, the Faculty of Nursing, the Faculty of Medical Science, the Faculty of Allied Health Sciences, and the Faculty of Public Health. The Science and Technology cluster incorporates 4 faculties: the Faculty of Science, the Faculty of Engineering, the Faculty of Agriculture, Natural Resources and Environment, and the Faculty of Architecture. The Social Sciences cluster is made up of 5 faculties: the Faculty of Education, the Faculty of Humanities, the Faculty of Social Sciences, the Faculty of Business, Economics and Communications, and the Faculty of Law. Our goal is to achieve academic excellence in order to produce well-rounded graduates who are academically competent, professionally trained, theoretically, practically, and intellectually updated with current global issues, and self-motivated. Naresuan University is committed to the 4 key missions, which include well-qualified students. The university produces graduates who are skillful and well-rounded in their professions. Research. Our research focuses on innovation and invention of technology to serve economic and social development. Academic services. The university contributes to educational decentralization in local communities. Arts and culture conservation. Conservation understanding will forge a sustainable mindset. Regardless of time, Naresuan University has a promising commitment to becoming an excellent university, producing highly capable students with good ethics and morals. This will ultimately bring Naresuan University to be one of the leading universities engaged with outstanding research as well as well-qualified students. This is what the university has to offer.
So that was the introductory presentation of Naresuan University. Thank you very much for your great attention.
So it's been 25 years since the establishment of Naresuan University, and we have served the community with the main missions to provide good academic services as well as establish academic networks. Therefore, various activities have been conducted to achieve our main goals, and one of it is the conferment of an honorary doctorate in various fields. Naresuan University has offered honorary doctorate degrees to distinguished people. For example, Dr. Mahathir bin Mohamad, the fourth Prime Minister of Malaysia for honorary doctorate in political science, and President U Thein Sein, the eighth President of Myanmar for honorary doctorate in political science. And what about people from science and public health fields?
Yes, certainly. In the fields of science and public health, Naresuan University has conferred honorary doctoral degrees on a number of distinguished people from around the world, namely His Majesty King Jigme Khesa Namgyel Wangchuk.
Is that the king of the Kingdom of Bhutan?
Yes, King of the Kingdom of Bhutan, for honorary doctorate in public health. Professor Sir Harold W. Kroto, Francis Epps Professor in the Department of Biochemistry and Chemistry, the Florida State University, USA, and a co-recipient of the 1996 Nobel Prize for Science for honor degree in science. So, ladies and gentlemen, this year marks a very special occasion that Naresuan University is becoming an active part of the ASEAN community, symbolizing a global connectedness. When we talk about globalization, we immediately think about health, the mechanism of health, movement of health, social, economy, and politics. The work of Professor Beutler signifies an importance of innate movements and mechanisms, and that is the innate immunity. His pioneering discovery has made such a significant contribution to our understanding of inflammatory and inflammation that revolutionized medical treatment. And this is why, ladies and gentlemen, this year, Naresuan University confers honorary doctoral degree on Professor Bruce Allen Beutler.
Now, ladies and gentlemen, we would like to take you to the conferment of an honorary doctorate in medicine on Professor Bruce Allen Beutler, who was awarded the 2011 Nobel Prize in Medicine.
First of all, may I take this opportunity to invite the following honorable people to be on the stage. Professor Dr. Grasset Chanawong, Chairman of Naresuan University Council.
Next, may I invite Professor Dr. Sujin Jinayon, the President of Naresuan University.
Assistant Professor Dr. Siri Kasem Sirilak, Dean of the Faculty of Medicine, Naresuan University.
And next, the Naresuan University Council members. And the last person we would like to invite to be seated on the stage is, ladies and gentlemen, please give a big applause to welcome our honorary doctorate and Nobel Laureate, Professor Bruce Allen Beutler. Please, Professor.
At this auspicious moment, may we take this opportunity to invite Professor Dr. Krasaya Chanawong, Chairman of Naresuan University, to be at the podium in order to read the citation for Professor Bruce Allen Beutler. Please, Mr. Chairman.
Distinguished guests, ladies and gentlemen, it's a great honor to have this opportunity to make a good citation for Professor Bruce Allen Beutler. Professor Bruce Allen Beutler is a 2011 Nobel Laureate for Medicine and presently is a director of the Center for the Genetics of Host Defense at the University of Texas Southwestern Medical Center in Dallas, the United States. He also holds the Raymond and Ellen Wiley Distinguished Chair in Cancer Research, and since 2011 is the Regental Professor of the University of Texas System. Professor Beutler was born in Chicago and raised in Southern California. His father, Ernest Beutler, was a physician and medical geneticist. From an early age, Bruce Beutler developed an interest in biology, spending long hours in his father's laboratory. Professor Beutler received his undergraduate degree in biology from the University of California, San Diego, after which he went to medical school at the University of Chicago, receiving his medical degree at the age of 23. He then did his 2-year residency in neurology at the University of Texas Southwestern Medical Center in Dallas, Texas. In 1983, he became a postdoctoral fellow at the Rockefeller University in New York City, where he was able to isolate the mouse tumor necrosis factor, or TNF. He was the first to recognize the importance of TNF as a key executor of the inflammatory response. In 1986, he returned to the University of Texas Southwestern Medical Center in Dallas, working as an investigator and professor of internal medicine at the Howard Hughes Medical Institute. He designed inhibitors of TNF that are widely used in the treatments of inflammatory diseases. Continuing his work on the immune response, he worked at the Scripps Research Institute in La Jolla, California, from the year 2000 to 2011, where he was a professor in the Department of Immunology and later chairman of the Department of Genetics. In the year 2011, he returned to the University of Texas Southwestern Medical Center, where he continues his work on host defense mechanisms. Before he received the Nobel Prize for Medicine for his discovery of an important family of receptors that allow mammals to sense infections when they occur, triggering a powerful inflammatory response, Professor Beutler was recognized by the Shaw Prize in 2011, the Albany Medical Center Prize in Medicine and Biomedical Research in 2009, election to the National Academy of Science and Institute of Medicine in 2008, the Frederick B. Bang Award in 2008, the Balson Prize in 2007, the Grand Prix Charles Leopold Mayer in 2006, the William B. Coley Award in 2005, the Robert Koch Prize in 2004, and other honors. He has authored or co-authored more than 300 papers which have been cited more than 46,000 times. Ladies and gentlemen, I'm proud to have the honor to make a citation and announcement on behalf of Naresuan University that Professor Bruce Allen Beutler has dedicated his professional life to the struggle against infectious disease, and his discoveries have been realized as drugs in the treatment of inflammatory diseases. For all the work he has done and for the contribution he is still making to the advancements of medical science, Naresuan University is honored that Professor Bruce Allen Beutler has accepted the university conferment of an honorary doctorate in medicine in 2014. Thank you very much for your attention.
Thank you very much, Mr. Sharman, for such a wonderful and touching, citation.
Ladies and gentlemen, I would like to invite Professor Dr. Sujin Jinayon, the President of Naresuan University, to confer the honorary doctoral degree in medicine on Professor Bruce Allen Beutler. Please, Mr. President.
Thank you very much, Mr. President.
Ladies and gentlemen, may I take this opportunity to invite Assistant Professor Dr. Siri Kasem Sirilak, Dean of the Faculty of Medicine, to present the academic gown to Professor Bruce Allen Beutler. Please, Professor.
Thank you very much, Assistant Professor Siri Kasem.
Ladies and gentlemen, please give another big round of applause to our honorary doctorate in medicine, Professor Bruce Allen Beutler.
In order to express our deep gratitude and appreciation to our Nobel Laureate, may I take this opportunity to invite Professor Dr. Krasa Chanawong, Chairman of Naresuan University Council, to present mementos to Professor Bruce Allen Beutler. Please, Mr. Chairman. The special mementos that were wisely selected for Professor Beutler today are, first, a silver plate with Professor Beutler's name carving on it. This silver plate is 100% handmade by the skilled artisans in Thailand. And it was specially designed as a gift for Professor Beutler.
And the second memento is the Naresuan University wooden trophy.
The third is the model of King Naresuan's armor. As you may notice that our university is named after King Naresuan the Great. So this is representing the great remarkable victory in freeing the kingdom of Thailand.
And the fourth memento is a pop-up book of King Naresuan biography. These are beautiful, aren't they? Mm-hmm.
Exactly. And that's the end of the conferment ceremony.
So please give another big round of applause to Professor Alan Beutler.
Ladies and gentlemen, I think we have come to the noteworthy and breathtaking moment that we have been waiting for. The program for part of the 5th ASEAN event series, Bridges, Dialogue Towards a Cultural Peace. Is that right, Dr. Chayanin?
Yes, Ms. Pongam. On behalf of Naresuan University, we organized this event as we believe that it would be an opportunity for you to establish possible linkage with the world-renowned figures in terms of staff development, research activities, and sharing of perspectives that may be of interest between you and Professor Beutler. And we are very delighted today that you have come to participate in such a conferment ceremony and in this lecture. Thank you very much for that.
Well, not only Naresuan University is the host for this event, we also have our co-host that must be mentioned. So may we take this opportunity to give a brief introduction about the International Peace Foundation as a co-host, please.
The International Peace Foundation has been established exclusively for not-for-profit purposes, in particular to support peace universities as well as scientific projects and institutions in dealing with research into conflict prevention and strategies for solution of conflicts. The foundation also promotes peace activities, understanding and social exchange between peoples, their cultures and traditions.
Thank you for that introduction. Before we are going to hear Professor Beutler's valuable lecture, I would like to invite Dr. Uwe Morawetz, Chairman of International Peace Foundation, to give us some background and introductions of the lecture. Please, Mr. Chairman.
Sawadee Krap, and welcome to the 5th ASEAN event series, Bridges, Dialogues Towards a Culture of Peace. Bridges is facilitated by the International Peace Foundation, a non-political and non-religious foundation under the patronage of 21 Nobel Peace Prize laureates based in Vienna. The events are hosted in cooperation with various local partners, including some of the country's major universities, and I would like to thank Naresuan University for hosting our event today. Having started in September last year, Bridges will now be continuously held in Thailand and Singapore until March 2015, involving the participation of Nobel Laureates for Peace, Physics, Chemistry, Medicine, and Economics. The 5th ASEAN series of Bridges follows the series of 500 Bridges events, which the International Peace Foundation has already hosted in Thailand, the Philippines, Malaysia, Cambodia, and Vietnam since 2003, as an independent contribution to the Decade for a Culture of Peace and Nonviolence initiated and promoted by the United Nations General Assembly. The pluralistic program of Bridges highlights the International Peace Foundation's intercultural and transdisciplinary approach towards peace. The foundation does not take sides, but acts as a mediator by creating independent platform for dialogue where representatives of science, politics, economy, culture, religion, the media, and youth can meet, share their viewpoints, listen to each other, and find mutual ways of understanding and cooperation. Therefore, the foundation itself is a bridge and a facilitator between different language groups in our divided societies, where politicians speak another language than artists, and business and religious leaders another one than scientists. In a highly interdependent world, problems cannot be solved by either one of these language groups only, but by working together. After the success of its bridges programs in Thailand, the Philippines, Malaysia, Cambodia, and Vietnam, the International Peace Foundation has been invited by other ASEAN countries to build further bridges of peace and international understanding by expanding its program in Southeast Asia to stimulate the intellectual, scientific, and cultural exchange in the region. The 5th ASEAN Bridges series therefore continuously takes place in Thailand and Singapore from September to March, comprising events with Nobel laureates from all fields. The Nobel laureates visit the region, not all at once, but separately, to conduct public lectures, seminars, workshops, and dialogues hosted by local institutions during a continued period of 6 months. The aim of Bridges is to facilitate and strengthen dialogue and communication between societies in Southeast Asia with their multiple cultures and faiths, as well as with peoples in other parts of the world, to promote understanding and trust. The events aim at building bridges with local universities and other institutions in Southeast Asia to establish long-term relationships with Nobel laureates in all fields, which may result in common research programs and other forms of collaboration. By enhancing science, technology, and education as a basis for peace and development, the events may lead to a better cooperation for the advancement of peace, freedom, and security in the region with the active involvement of the young generation, ASEAN's key to the future. This is why Bridges is not designed as a one-time event, but as a series of synergies to make the events a sustainable success for Thailand, for Singapore, and for Southeast Asia as a whole. I am grateful to Naresuan University as well as to our other partners and sponsors who have enabled us to make the idea of Bridges a reality. I would like to thank everyone present today for taking part in this program. May it help us to facilitate a new culture of peace through dialogue, transcending its definition as merely the absence of peace, of war or armed conflict, into a new understanding what the basis for peace is: education. In this spirit, we welcome today the 2011 Nobel Laureate for Medicine, Professor Bruce Beutler, who has agreed to come to Thailand to support the events. We all look forward to his keynote speech and to his important contribution to build bridges. A warm welcome, Professor Bruce Beutler.
Thank you very much, Mr. Chairman, for your perspectives and special introductory speech.
And Dr. Morawetz has a memento that he would like to present to Professor Beutler.
Because we have, where is it? A very special golden elephant. Kindly sponsored by Kunprida and the Pranda Group. Thank you for this. So may all your memories of Thailand be golden. Thank you very much.
Lovely.
Lovely. And I think everyone knows that, the elephant is the symbol of Thailand, right?
Yes.
So that's a very lovely present indeed. Thank you very much. Again, Dr. Xianin, could you please tell us a little bit more about Professor Beutler?
Certainly. Professor Beutler has written a very inspiring biography upon receiving his Nobel Prize in Medicine in 2011. He mentioned his father as a role model, that he was very lucky to have an understanding father who was himself a distinguished scientist. The path to Nobel Prize was an industrious one. Professor Beutler confidently pursued a career in biology, and you would be amazed to know that his principal achievements resulted from addressing a single problem with relentless obsession, disregarding any doubts and discouragement. And in the event of failure, one may always pick another problem and try again.
Wow, that's very inspiring. And now, ladies and gentlemen, I think we are ready for the long-awaited lecture. So we both would like to pass over the floor to the 2011 Nobel Laureate for Medicine and invite him to deliver his lecture on the global struggle against infectious disease. Ladies and gentlemen, please give a big applause to welcome Professor Bruce Allen Beutler. Please, Professor.
Well, thank you. I'm practically speechless now just when I need to speak. But I will speak. I want first to say how grateful I am at my reception here, and also how thankful I am to Naresuan University for this tremendous honor. I'm really deeply touched by it, and I'm very happy to be here today. We're getting ahead of ourselves in the talk, so let's go to the beginning instead. And let me just make a few comments about the title and why we're talking about a global struggle against infectious disease. I think you'll see in the course of my talk, which takes a somewhat historical approach, that we've all been here before, that this is a problem solved by one part of the world after another. And as communications have grown in recent times, we've come to see that it is indeed a global struggle. No one is immune to infection when it crops up in one part of the world. It's rapidly transferred to another. And the best of technology in certain parts of the world has to be coupled with the efforts of local populations and brought to bear to solve this problem if we're really to progress as a civilization, a global civilization on the earth. I'd like to tell you also that it's my personal view that science has very much made the world that we live in today. It's very much shaped how civilizations have developed, in some cases how they failed to develop. And as you'll see when I speak to you and give you a few examples, in the area of immunology, in the area of infectious disease, this has been particularly prominent. We'd really be nowhere at all if not for certain areas of progress in science related to immunology. The numbers tell the story very well, and I can begin with a few statistics for those among you who keep track of such things. If we look at this chart, we're going to be looking at median lifespan. So you see the red line is the line indicating the time at which half of the population has died. And it may come as no surprise to you that in the United Kingdom, in the year 2000, the median lifespan was close to 80 years. And it's probably even a little bit beyond that now, and it's not so different from the average lifespan. In the same year, the situation was very different in the country of Mozambique. In Mozambique, 50% of all people had died by the age of 39. This is a somewhat shocking statistic for someone who grew up in Southern California and considered life a permanent state of affairs from childhood on. And yet those were the numbers, and we can understand very well why they're that way. A real surprise comes when we look back at the year 1860 in the city of Liverpool, England, where vital statistics were kept and well documented. And we see that the situation then in Victorian England was even worse than in Mozambique in the year 2000. Surprisingly and somewhat shockingly, 50% of all people were dead by the age of 10 in Liverpool at that time. Maybe it's surprising also that if you go back further in European history to Breslau, 1690, or to Neolithic times based on records told by bones, or even to Paleolithic times, the situation never got much worse than that. In Victorian England, people were living pretty much in the wild state when it came to confronting infectious disease. And you can be sure, though I won't go into detail, that most of this attrition in life was due to infection. Now, one thing you have to keep in mind here is that our tremendous progress from here all the way up to here is something that didn't evolve. It's not that we have better innate immunity or adaptive immunity now than we did then. This is intellectual progress that made the difference. It was the human brain, the collective human brain, if we talk about that, that made humans begin to live about 8 times as long, in the median case, 150 years after the year 1860. In other words, we really changed ourselves as a species, and we did it by public health measures, by immunization, by all of the methods that are probably familiar to you. Infections have diverse effects. They've been known since antiquity, but they have common themes as well. They're transmissible diseases. They all have inflammatory characteristics, and if we look at them all together, we can say, without a doubt, they've stolen more lives and more years of human life than any other cause. More than famine, more than war, more than lions on the African veld, the real thing has been infection. Smallpox alone, before it was eradicated, is believed to have claimed about 1 billion human lives. And in the 20th century, when things were well documented, the number is placed sometimes on the higher end at about 500 million for that century alone. Imagine that it had not been eradicated. In all, looking across the world today, about 1/4 of all people born will ultimately die of an infectious disease. Now, this is not the greatest killer of human beings. On the planet, we can say that cardiovascular disease kills more people, but infectious disease tends to kill people at an early age, and because infection does that, it stands as a tremendous selective pressure, and it's come to shape our genome dramatically. It brought an immune system into existence long, long ago, and even in recent times, it continues to influence the frequency of certain alleles of certain genes. What has this selective pressure done to our species and to all species? Well, we've only known about microbes as a cause of infection for about 150 years, but the battle certainly has gone on for 1 billion years or more. And we have to ask ourselves how the immune system evolved. That's been a question of constant interest to me. We imagine, most of us imagine, that it evolved in a very simple way, that probably antimicrobial peptides were elaborated by microbes in order to combat other microbes. And we still see examples of this today with antibiotics like bacitracin, for example, which are peptides, which are cytolytic, and there are many, many other examples. But it's expensive to maintain a constitutive apparatus for fighting microbes around you, and for that reason, probably sensing and signaling components had to evolve early on as well. As multicellular organisms developed, we know that they developed specialized cells for immunity, things like macrophages that could engulf microbial invaders and destroy them, and that was probably the birth of innate immunity, which persists to this time in many life forms, including ourselves, and is essential for survival. Components of a sensing signaling apparatus seem to have predated the split between plants and animals. And we know this because we see the remnants of common domains used for immune function in proteins that exist today. Things like the leucine-rich repeat domains of toll-like receptors, tear domains of toll-like receptors in other molecules, and nod-like receptors exist in both. And here are some examples of that. This is a molecule that I'll have much to say about later on, or these are rather. These are toll-like receptors. This is a plant disease resistance receptor. They both have leucine-rich repeat motifs that are excellent binding motifs for foreign molecules. They both have tear domains, some represented in the cytoplasm, as in the plant, others attached to the toll-like receptors. There are protein kinases of a similar shape in both cases. There are adapter proteins that exist in both cases. And I could go on, but that's just the evidence that the split occurred very, very early in evolution. The evolution of immunity then is ancient, and we have to think what it was like in the early days of animal and plant life and what sort of immunity may have developed after that. Animals are believed to have come from dinoflagellates, which are motile organisms, and plants followed a very different path, of course, in their evolution. But perhaps 550 to 500 million years ago, something extraordinary happened where animals were concerned. And everyone who's interested in evolution would have loved to be alive at that time. It was known and is known as the Cambrian explosion. At that time, in the shallow, oxygen-rich seas of the Earth, a fantastic variety of animal life developed in just a short period of time. Nobody knows why there was such a prolific expansion in the different lines of animal life. Perhaps because there was nothing before to fill many niches that existed waiting to be colonized. But the point was that organs developed that hadn't been present before. A segmented body plan developed, eyes developed, in some cases teeth developed, limbs and everything that we know in modern animals today. We can trace its origins back to the Cambrian explosion. Out of the Cambrian explosion came all of the familiar animal forms of life that we know today. We have the vertebrates, and the vertebrates exist in the form of jawed and jawless vertebrates. We have also the cephalochordates, urochordates, then echinoderms, mollusks, insects, and all of these life forms. It's rather shocking to me to see that plants were a bit retarded in their development, and it wasn't until practically yesterday that flowering plants developed, the monocots and dicots that seem to have taken over the world and with which we are so familiar today. Where immunity was concerned, something special happened in the vertebrates, really something almost miraculous. Vertebrates developed a special plant for making receptors unlike any other. Vertebrates developed a technique to make receptors that could recognize any molecule that existed in the exterior world or any molecule that ever could be. In the Cambrian explosion, a new cell form was born, and that we call the lymphocyte. It is rather unassuming looking. It has a small rim of cytoplasm and a large smeared nucleus, but these cells, which existed a little bit before immunity developed, were to play a huge role in immunity later on. They're made in organs like the typhlosol, in the jawless fish, in the thymus, in the bone marrow, in birds in the bursa fabricius, and they all look about like that. Now we know that before an immune function was acquired, there were already B cells and T cells. And we know this because both lineages were transmitted into the jawed and jawless fish lineages. But where jawless vertebrates were concerned, a different substrate for producing receptors evolved. The receptors were enabled by the transmission, probably horizontal transmission, of recombinase genes from bacteria, RAG1 and RAG2. And at that point, these enzymes began to operate on different parts of the genome, and by recombining parts of the genomic DNA in jawless vertebrates, they made a set of receptors, highly diverse receptors, that were made out of leucine-rich repeat proteins, whereas in the jawed vertebrates, immunoglobulin-type repeats were favored, and they made what we call today antibodies and T-cell receptors. This is adaptive immunity. It developed 500 to 550 million years ago, give or take. We don't know exactly when. And that is one legacy of our struggle against microbes. But it has a legacy all its own. The legacy of having such a sophisticated immune system is that you can develop autoimmune diseases. Rheumatoid arthritis, gout, type 1 diabetes, systemic lupus erythematosus. These are all examples of autoimmunity. It's believed that something like 12 to 14% of us will develop one or another autoimmune diseases in the course of our lives, and this by itself is a considerable medical burden, as you can imagine. There are also more subtle forms of inflammation. Many today would say that arteriosclerosis has a kind of inflammatory basis. Certainly monocytes and other inflammatory cells collect in atherosclerotic plaques, and they're probably important to the pathogenesis of disease. Neurodegenerative diseases like Alzheimer's disease may have even more subtle forms of inflammation that are pathogenically important. So depending how broadly you define inflammatory disease, it could be said to affect almost all of us at one level or another. But let's go back to infection and back to Victorian England. At that time, these were the dreaded pathogens of the age: smallpox, typhoid, tuberculosis, and cholera. They were known not long after 1860 to be caused by microbes, and people made attempts to deal with them even before there was that knowledge. This is a classic painting of Edward Jenner, a physician who took cowpox lesions from Sarah Nelms, shown at the right, and inoculated them into James Phipps, a somewhat unwilling patient sitting in the chair. And he was able to demonstrate that this new procedure, which was called vaccination, to refer to cattle as the origin of the inoculum, proved to be very effective in preventing smallpox, much more effective than variolation, which had preceded it, where people would actually inoculate with live smallpox and hope to prevent a more serious disease later on. Then, as now, there were people who criticized this approach. There was an anti-vaccine society that sprang up in the 19th century. People were afraid to receive this therapy, yet imagine if it had not been implemented widely. And for a good long time it wasn't implemented widely. It certainly would have prevented the progress of civilization. The problem was that in the time of Jenner, and for some while after, all approaches to infection were simply hampered by ignorance of what infection was. Microbes were known from the time of von Leeuwenhoek, and yet the link between infection and microbes hadn't been made, even though it seems second nature to us today. Until it was made, there were only vague ideas about how disease was transmitted between individuals and how disease seemed to produce effects that were akin to poisoning. The kind of arguments that went on favored either miasma as a course of disease. In other words, miasma was a cause of disease that was somehow involving air, the transmission through air, fetid air in general, and contagion. Advocates of the contagion hypothesis held that you had to have direct contact with the infected person in order to become infected yourself. Scientists of the time did begin to look at this matter, and noting that infection looked a good deal like putrefaction of organic material when observed, as in the case shown at the top, they began looking at fetid meat and vegetable products, grinding them up in hopes of finding a toxic product that might convey something similar to infection. In the 18th century, Albrecht von Haller, and in the 19th century, Francois Magendie, a great and famous physician, began doing experiments of that kind, and they found that putrid materials of plant and animal origin would elicit fever and sometimes death in animals to which they were transferred. Later, people became a bit more sophisticated, and Ernst von Bergmann, Theodor Billroth, and Peter Panum began actually trying to purify the toxic principle using the tools that were available at the time. Peter Panum got quite far with this, and he actually found an alcohol-insoluble material, 12 milligrams of which was sufficient to kill a large dog. and we know today that that material was probably what we call lipopolysaccharide or endotoxin, based on Panum's description. But the real heyday of success against infection began with Louis Pasteur and Robert Koch. They founded, both of them rather independently, the germ theory of infectious disease. And from that point forward, rather than starting with something like putrefied meat, one could actually look at pure cultures of microbes and try to understand what was toxic about them, what was recognized, what could be responded to by the immune system. One problem with these two was that they hated each other. They absolutely hated each other, and this had a good deal to do with nationalism, with antagonism between France and Germany. It was a fact that Pasteur's son was either injured or killed in the Franco-Prussian War, and he didn't like Germans very much for that reason, and certainly there was the element also of professional competition that spurred him on, and Robert Koch reciprocated. It's kind of amusing to read today the letters that they wrote to one another. I will read aloud. In his Geneva lecture, Pasteur bitterly complained about my having rejected his microscopic examinations and inoculation techniques, wrote Koch. However, after his inoculations with saliva and nose slime and his repeated discovery of the microbe en wheat, I am not able to change my opinion. Pasteur deserves criticism not only for his defective methods, but also for the way in which he has publicized his investigations. I have to preface for you here that Pasteur had presented a lecture in which he claimed to have found a new microbe in the froth that was emitted from the nose of patients suffering from rabies, but he didn't say that this was the cause of rabies, and Koch, by design or perhaps by real misunderstanding, thought that he had said that he had found the cause of rabies. And Pasteur replied, I find here, monsieur, a new example of the manner of discourse that has served you previously in 1881. You attribute to me some errors which I hadn't committed, you refute them, and then exult noisily. So it is a pity that they didn't get along, the French and the German microbiologists. I like to think that that wouldn't happen today, yet still it does from time to time. There definitely is animosity among scientists, and it's certainly to be avoided if possible. Nonetheless, for all this impediment, one after another, infectious diseases were ascribed to microbes during the 19th and 20th century, and the germ theory of infectious disease became very widely accepted. And then almost immediately a new question arose: how is it that germs do injury to us, and how is it that we detect them and begin to mount an immune response? These were questions partly of immunology, partly of microbial pathogenesis. Among the students of Robert Koch were Ehrlich, who was well known for his identification of antibodies, von Behring, who worked with him and worked on the solution to diphtheria, making diphtheria antitoxin. And there was Richard Pfeiffer, probably the most obscure of the three, but he is the one who founded the field that I later became involved in. It was Richard Pfeiffer, who was a military surgeon billeted to Berlin in the year 1891, who founded the endotoxin model of toxicity from microbial infection. It was Pfeiffer who started to work with cholera and noted that heat-killed cholera vibrio were lethal to guinea pigs if injected in sufficient quantity. And he attributed this to what he called a toxin in the body substance of the bacterium, rather than on the surface, as we now know it to be. And this material, which he called endotoxin, would cause fever, inflammation, shock, sometimes vasomotor collapse, and death in guinea pigs. We fast forward now a few decades. We know today that endotoxin, or lipopolysaccharide, is on the surface of gram-negative microbes. It's based on both a polysaccharide moiety and a lipid A moiety, and it's a major structural component of the outer membrane of gram-negative bacteria. By the 1970s, the structure of several endotoxins had been solved quite rigorously, and by 1984, a lipid A molecule had been synthesized completely artificially in the laboratory in Japan and also in Germany, and these molecules could do just about everything that Pfeiffer had seen nearly 90 years before. We might draw a typical LPS molecule this way. It has what we call a KDO. It has a ketodeoxyoxylosonic acid, a polysaccharide chain trailing off into the medium, and then these acyl chains with a characteristic acyl-oxyacyl linkage. Pfeiffer, during his lifetime, was nominated to win the Nobel Prize 33 times, but he never received it. Nonetheless, the reason for his nomination was quite clear. Every day, hundreds of people die of endotoxic shock. This is a child with gram-negative meningococcal sepsis. It shows many of the features of endotoxic shock. People begin to bleed uncontrollably. They have a stiffening of the lungs and require intubation. The kidneys and other organs are damaged by endotoxic shock, and there's hypoxia and need to be ventilated artificially. You see some blood also in the nasogastric tube, also a problem. If it comes to this stage, maybe a 50% mortality might be expected. It's also the case that endotoxic shock is a severe systemic form of inflammation, and in those days, as until very recently, the origins of all inflammation were unknown. We could guess that this might be biochemically and at a cellular level rather similar to the kind of inflammation that occurs in non-infectious diseases, yet no one knew where it started. Nearly 100 years went by, and no one was able to find a receptor for endotoxin, although the concept of receptors was well entrenched in biology during the 20th century. I personally began to be interested in this problem about 40 years ago when I worked in the laboratory of this man, Abraham Braudy, who was interested in making antibodies against LPS to protect against endotoxic shock. And undoubtedly, I first heard the word endotoxin then when I was about 16 or 17 years old. I was also interested in microbiology and in the tales of the early microbe hunters. I read The Microbe Hunters by Paul de Cruyff. I read a book by Sinclair Lewis called Aerosmith about a young doctor and his struggle against infection in the early 20th century. Sinclair Lewis did win a Nobel Prize, I have to point out, for literature. And I was inspired by these books, and they undoubtedly shaped my decision to go in the direction that I did later on. Because of those books and because of my father's advice on the subject, I decided to go to medical school, and afterward I did a residency and internship at UT Southwestern. And then I had had enough of clinical medicine for a time, and I decided that I would go back to basic science, which was really my interest all along. I joined a laboratory in New York City where there was an interest in wasting in chronic disease. This is a cow in Kenya with trypanosomiasis, and it can be imagined that there are probably only a few grams of the parasite in the entire animal, and yet you see it's wasting away, it's lost its appetite. We see this in tuberculosis, in many chronic diseases. And the question was why? I set up an assay system to look at the question from an immunologic standpoint. The idea was that perhaps immune cells recognize something made by the infectious organism, and they make a factor which we called cachectin, which in turn interacts with the energy storage tissues of the body so as to cause a failure to take up fat from the exterior medium. It was believed in some variants of this hypothesis that other things might also trigger such a response, endotoxin, perhaps things made by malignant tumors. And in the end, when the matter was looked at closely, it seemed nothing worked as well as endotoxin to induce cachectin activity. The assay for cachectin activity was simple. I would produce cachectin by stimulating mouse macrophages with LPS. I would apply that to a cultured line of fat cells, and I would measure suppression of lipoprotein lipase, an enzyme needed to take up exogenous fat. Using this assay, I purified a single protein species, and we called it cachectin at the time, but not long after I found that this same molecule was homologous to human tumor necrosis factor, recently isolated by an entirely different approach. As the name suggests, that activity had to do with the ability to lyse tumor cells in vitro. So here was something very curious. It was really the start of my career. It seemed that this single molecule, TNF, made by macrophage in response to endotoxin, would destroy tumors, it would cause the resorption of fat. These were both things that LPS could do, and I wondered, could this one molecule do everything that LPS could do? It didn't take long to see that it would cause mice to become very ill and even to die if more than 20 microgram were administered to them, where a denatured preparation of TNF wouldn't have that effect. And furthermore, if mice were immunized against their own mouse TNF and then challenged with LPS, they could better survive the LPS. The dose lethality curve was shifted to the right at the 50% mortality point, and this convinced me that this was at least one major mediator of endotoxic shock. So now we'd moved beyond putrefaction caused by miasmas or contagions. We'd gone beyond bacteria, we'd gone beyond LPS, and we had a mediator of injury caused by LPS that was made by the host. And it was this mediator that I hoped to follow in order to understand what the LPS receptor might be. It seemed the ideal endpoint because it really did mediate much of the toxicity of LPS. So here was the question. By the year 1990, it was known that LPS somehow required CD14, a cell surface protein found by Wright and Ulevich, to be necessary for the LPS response. Yet this couldn't really be the signaling receptor because it had no cytoplasmic domain, couldn't signal into the cell. There must be a co-receptor, most people thought, that would mediate that event. And this receptor was a complete mystery. Whatever it did, it must activate NF-kappa B because the TNF gene had NF-kappa B motifs in its promoter region, and it had to be activated in that way to produce first the TNF mRNA. That RNA existed in a locked, untranslatable form, and it must be unlocked by a second signal from the receptor, and then one could get secreted TNF, processed and secreted TNF that would do what TNF does. The central mystery here then was, what is the LPS receptor? If we could find that, we might understand how microbes are detected by the immune system. There were many ways to go after the receptor, and I would say we tried about all of them, but the one that worked was a genetic approach. And the answer to the question lay in two substrains of mice that wouldn't respond to LPS because of spontaneous mutations. The existence of these mice suggested that LPS was sensed in a very specific way. It didn't just integrate into membranes at random. It depended on a certain protein. But which protein and how to find it? These two strains were the C3H-HEJ strain, which had been known to be LPS resistant since 1965. It was known, in our hands anyway, to fail to make a cytokine response to LPS, and only to LPS. Other things like nucleic acids would activate the cells. In 1978, the mutation was mapped rather roughly to a point between two visible loci by Watson and Riblet. These loci were on chromosome 4, and their positions isn't known even today. In 1978 as well, another strain of mouse, C57 black10 SCCR, was found to be resistant to LPS by Antonio Coutinho, and he crossed the two strains and he found that the same gene was involved. The offspring were all strongly resistant. Now, how to track down such a gene? It was a difficult matter in those days. one knows that in the mouse there are 20 chromosomes plus the sex chromosomes, and each one is packed with chromatin. In chromatin structure, you have DNA that's wrapped tightly around nucleosomes, and the DNA itself is a long string of base pairs, and if you hope to find a mutation that may be caused by a single base pair change, you may have to search through 2.7 billion bases of DNA to do it. That was the challenge, and yet we were really quite passionate about tracking that down. Fortunately, there is a way to exclude much of the genome. That's a process called genetic mapping, and I won't go into it in detail, but one has to come to a point where one proves by a kind of geometric reasoning that the gene of interest or the mutation of interest is between two flanking mutations on that chromosome, two markers. And then one has to clone all of the DNA spanning those two markers, and then one had, in those days, to search it for its content of genes. This was a backbreaking task. It involved sequencing largely by hand, blasting the sequences with a computer to look for expressed sequence tags that might match the genomic DNA. And day in and day out, we kept doing that for a period of about 3 years. By August of 1998, about 90% of the critical region had been thoroughly explored for genes, and we had found only pseudogenes. Only, of course, they didn't come labeled with a note that said, I'm a pseudogene. We had to prove in every case that they really weren't the gene we were after by comparing the mutant and wild-type control strains. All at once, in August of 1998, I was blasting as usual in my study one night, and I found a very powerful hit, much better than ever before. Then another hit a few minutes later, and I felt surely we had found the gene this time, because these were the best quality hits I had ever seen, and also we'd explored almost all of the region, and there just wasn't much space for any more genes to exist. There was still another reason for excitement as I looked at the gene and what it encoded. I mentioned to you that CD14 was one component of the LPS receptor complex, and it was a leucine-rich repeat protein, and so was this new item, TLR4, that I had found encoded in the contig. But this protein had a membrane-spanning domain and a cytoplasmic domain similar to that of the interleukin-1 receptor, a protein with known inflammatory potential. It made sense to think that LPS might be transferred from one molecule to the other, and that the signal would be elicited by the tear domain, as it was called, that might activate NF-kappa B. There was another reason to believe that this was the right gene. Earlier work by Jules Hoffman had focused on the toll pathway in Drosophila, formerly known for its developmental effects, and he had shown that in the fruit fly, toll and its ligand, Spätzle, were necessary for an effective response to a fungal infection. Inoculate flies with Aspergillus fumigatus, and if they lack either of these two genes, they're overgrown by the fungus and killed. This situation was similar to that in the mice, which were intolerant of gram-negative infection and could be killed by small inocula. So I thought we probably had an evolutionarily homologous pathway. Now, in LPS-resistant mice, there were mutations that destroyed toll-like receptor 4, a point mutation in the HEJ strain and a complete deletion of the locus in C57 black 10 SCCR mice. And finding that was probably the highlight of my career. It was something that we hardly could believe when we found the mutations, and we knew we had solved a problem that had lasted almost 100 years. We also were able to deduce that this really was a physical receptor for LPS, and I won't go into that, but we had a genetic means of saying so. And it was later established that there was still another protein component of the receptor, a small molecule called MD2. 10 years went by before this complex was crystallized, and today we can see the structure. It looks something like this. You can see that you have the backbone of TLR4, this leucine-rich repeat domain, You see a small magenta basket, that's MD2, and the lipid A fits into that basket. A conformational change results, and that triggers endotoxic reactions. I've estimated that something like 0.1 nanogram of this complex is all it takes to mediate the lethal effect of LPS in a mouse. Time went by, and one was able to see the structures of several other toll-like receptors, not our work, but that of crystallographers. They all directly bind relatively conserved molecules of microbial origin. There are, in humans, 10 toll-like receptors, in mice 12, and in both species together 13. This is where recognition of microbes largely begins, though not the only system for that to occur. On that very first night of finding TLR4, we began to do some evolutionary studies. We found that the tear domain was the most conserved part of the molecule, and we made trees to look at the inheritance over evolutionary time of these structures. And what shocked me the most was not that only humans and flies had tear domains and toll-like receptors, but even plants had tear domains, and wherever they were seen in plants, those domains conferred resistance to infection. In this case, we're looking at a flax plant and a fungal rust. So truly, this was an ancient system devised for protection. In the long run, we'd like to understand how toll-like receptors signal. We'd like to understand the whole immune system, because we're machines and the immune system ultimately is a machine. And we've used genetics as our tool to do that. We induce mutations in mice at random, and then we track them down just as we track down this mutation, only we can do so much faster nowadays. At present, in our laboratory, it doesn't take 5 years anymore, but tracking a mutation down is a real-time process. If you find a mouse with an immune deficiency, within perhaps 1 hour, you know the cause of that immune deficiency. Now, I haven't time to discuss that, but I'll just say that while I was at Scripps, over a period of about 10 years, we were mutagenizing mice and looking at toll-like receptor responses, at TNF production once again. And everywhere you see a box here with red lettering, those are a collection of our mutations. And we were able to build quite an elaborate picture of how signaling works. It's complicated, and I won't go through it in detail, but I'll just show you that, again, the structural biologists have been at work here. And we can see in 3D now all of the molecules that participate in signaling, many of which are labeled by our mutations. And we can begin to put together quite a strong mechanistic picture of how signaling works, though I have to admit that some of the interactions here are real and some of them are still just fanciful. I have no doubt that in the next decade or so, we'll understand this about as well as we do a pocket watch, as far as how it works. That's the goal, and I think it's a realistic one. So what does all this do for us? Well, first of all, now we know how we see infectious microbes during the first minutes and seconds after they're inoculated. We might think we could mitigate the intense inflammatory response that actually kills people when they have a serious infection if we're able to keep that infection under control with antibiotics, and we often are able to. Many of you know that there are also a lot of immune deficiency diseases that are caused by failure of immune sensing. This is the famous bubble boy, and he had a problem with adaptive immunity, but there are people no less badly off than the bubble boy who have innate immune defects. And at last we have a hope of diagnosing such people because we know which genes are involved. I'd like to think that we can make better vaccines, having the knowledge that we do now about how the innate system is activated and how that transfers to an adaptive immune activation. Vaccines that are more efficacious, have lower toxicity, and are just better for specific purposes. And remember what I told you before about the legacy of our immune system being autoimmunity. That's unfortunate but true, and yet now we can understand how some of it comes about. most particularly in the case of systemic lupus erythematosus, we've gotten some insight that toll-like receptors are involved. And this is largely from the work of Anne Marshak-Rothstein. Just in very broad terms, we believe that in such a disease, there's inappropriate cell death for what reason we don't know. And as a consequence, nucleoproteins, RNA protein and DNA protein complexes are released. We all have B cells that recognize such complexes, all of us do, and yet usually our B cells remain quite quiescent. It's to these receptors on B cells that the proteins bind and they become internalized, and in the endocytic environment, there's acidification, and then there's recognition by toll-like receptors, either by the TLR7 or TLR9 complex. That gives a signal for cell proliferation. And where there may have been just a few such B cells to begin with, soon there are many of them, and they may differentiate to make plasma cells and memory B cells. The plasma cells will make more antibodies against nucleoproteins, and those will feed back to stimulate the memory B population and cause even more clonal expansion. And this goes on endlessly, and the end consequence is deposition of immune complexes in the tissues and an autoimmune disease. We can see this model operates very clearly in the mouse. Whether it's possible to interrupt it in humans at the level of TLRs is questionable, but it's certainly worth trying. I'm going to conclude now with some thanks to my staff. The work that led to the Nobel Prize for me was done by a pretty small group of workers. I'm not showing all of them, but the most critical ones would be Betsy Layton, Alexander Poltorak, Christoph van Uffel, and Irina Smirnova. Things change after you get the Nobel Prize. Now there are 62 people in our group, and this picture is already a bit outdated. The most exciting thing we're pursuing these days is the automated positional cloning of newly induced mutations, which may let us find all the components of the innate and adaptive immune systems before long. Again, I want to thank you all for this tremendous honor, and I'll be glad to have a dialogue with you and answer any questions you might have.
That was an insightful and marvelous lecture indeed. Thank you very much, Professor Beutler, for such a splendid lecture.
Ladies and gentlemen, another big round of applause for Professor Beutler, please.
Dr. Chianin, I saw you took notes seriously while you were listening. So which part of the lecture do you like most? For me, I like the very first part of his presentations when he said, your life longevity is depending on where and when you were born. So I just hope that I could live my life as I made it in Thailand, happily and long enough.
Yes, for me, I love the crystal structure of toll-like receptor.
That's very scientific,
yes.
right?
It reminds me of when I was reading biochemistry during my undergraduate years, and it is an honor for me to be listening to the, the founder of TNF-alpha and toll-like receptor, of course, yes.
Okay, so now I think it has come to the question and answer sessions. So I would like to give the floor to our audience who eagerly want to ask some questions. I think this is a very rare and very great opportunity for people who are interested or who are studying or working in this field.
So any questions are all welcome. Please just raise your hand and we will hand you over the microphone, or you can come to the aisle. Please do state your name and where you work or where you study before your question, please. Thank you.
As Dr. Chianin said, okay,
yes,
welcome.
We have a question over there.
Good.
Good afternoon, sir. I'm Ms. Piachat Rumngam from Medicine University, Thailand, from Department of Immunology. May I ask the question that could you please give us about your opinion in new trends or new strategies or new hot issue in cancer immunology, especially vaccinology and immunotherapy? Thank you.
Yes, thank you for the question. Cancer immunotherapy used to be kind of a distant dream. People tried to realize Ehrlich's passion to make magic bullets by tagging antibodies with toxins or with highly radioactive molecules, and I must say that didn't really succeed very well in terms of targeting tumors. In any case, one would like to have active immunity against a tumor, and there is a feeling that only with such active immunity can one truly eradicate a malignant disease. Now, it has been shown in recent years by investigators in several laboratories that there are inhibitory molecules like CTLA-4 and PD-1 that tend to put the brakes on immune response. And if one can inhibit those to the right degree, then the mutations that are present in cancer cells can be recognized sufficiently by the adaptive immune system to create a powerful response. It's a little bit early days. There have been some people in whom there have been complete remissions. These have been in large part in adenocarcinoma of the lung, renal cell carcinoma, malignant melanoma, and those latter two have always behaved in a little bit funny way, in the sense that there were occasional rare spontaneous remissions of those tumors. Adenocarcinoma of the lung is a real surprise, and if that holds up for a period of time, then I would say there's a bright future to trying to block those pathways and several others, and to use that approach to harness our own immunity to overcome cancer. It's a tall order when you think about it. Cancer has always been hard to combat with chemotherapy or with immune therapy because it's so similar to our own cells. Yet maybe the distinction is great enough, and that would be an enormous advance. And perhaps it would push the longevity curve out a few years more by itself.
I'm Dr. Budin, internal medicine staff from Faculty of Medicine, Naresuan University. This is my question. In Thailand, we are experiencing superbugs problems or antibiotics resistance of bacteria. A part of this problem is due to overuse antibiotics among our health professionals. What are your professional suggestions to relieve this serious problem? Thank you.
Well, thank you for that question too. It is a serious problem, and it's a global problem. It's the kind of problem that should concern everybody on the planet. We've been protected in this world for about 70 years with antibiotics. Before that, we had nothing. It's not clear that they're going to be good forever. It may be that the bugs eventually will win, or it may be that we, with our ingenuity, can chemically engineer antibiotics and always keep a step ahead of them. It'll be an interesting few decades to see which way it goes. But for the moment, the best approach to superbugs is education of physicians about what antibiotic to give. There is too little reliance on experts in infectious disease medicine. Doctors in the United States, and perhaps here as well, tend to simply prescribe medicines on their own, often inappropriate ones for the disease. Either the diagnosis is not right or the choice is not right. Culture and susceptibility may never be sent for, so one doesn't know what the best antibiotic would be. And this is what leads to disease resistance and to multi-drug resistance. So I think that's the area in which we have to concentrate, where we can make some progress right now.
Thank you.
Okay, thank you for the two questions. Yeah, by the way, any further questions?
Professor Beutler, thank you very much for your wonderful lecture. I have amazed by your lecture about TLR, that among plant and animal, TLR receptor and system
are highly conserved. My question is, why? And number two, both of them are worlds apart, yet they can direct or can be, sensed by another different species of from another world, bacteria. Evolutionary question, please. Thank you.
I believe the answer to that question is going to involve some speculation, as evolutionary reasoning always does. All I could say is that the binding function of leucine-rich repeat proteins proved to be very good, and we see that it was used later for binding things in the jawless vertebrates. I didn't mention it, but also things like ribonuclease inhibitor, a cytoplasmic protein that we all have, that binds ribonuclease very, very tightly with something like 10 to the 16th affinity constant. So this was the substrate that was available in the primeval soup before plants and animals differentiated 1 billion years ago. And it became so important to defense that neither of the derivatives ever let go of it, neither the plants nor the animals. They did develop other means of detecting and defending themselves, but those core mechanisms have stayed the same probably all of those billion years, and they've gradually changed to recognize different molecules. LPS, for example, isn't recognized at all by insects, so far as we know, not by the fruit fly anyway, but it is recognized by vertebrates and especially by animals. Evolution has changed the targets, but we've kept those same molecules all this time.
Another question, please? Yes, sir.
My name is Nusara Satpratpai. I'm from Department of Medical Sciences at the Ministry of Public Health. And first of all, I would like to thank for your talk today. It's very I very admire you because I take the toll-like receptor research as my PhD thesis. So you've always been my science idol. And I'm very thrilled to be here today. My question is about the dilemma of this innate immunity. As you know, having it is essential to us, but having it too much, we get autoimmunity. So nowadays, we have a lot of immunotherapy. And how could you find the balance between having it too much or having it too little? And could you elaborate more on that? That would be very
I have to fall back on evolutionary reasoning again and to say that, well, clearly toll-like receptor 4 didn't evolve to give us endotoxic shock. That doesn't make very good sense. One might say, well, maybe it evolved to weed out infected members of the herd and to prevent the transmission of disease by eliminating members of the herd. But I doubt that as well. I think without TLR4, an individual has diminished fitness. This is a system that evolved to work in the microenvironment. If you should happen to stick yourself with a thorn and introduce a few gram-negative bacteria, that's when the system performs very well. It will create local inflammation, it will attract the attention of the adaptive immune system, and it will contain the infection before it becomes widespread, before you develop sepsis. That's the balance that evolution seems to have struck between local action and systemic action. It has to be strong enough to marshal responses beyond the immediate wound, but not so strong that it kills the host. That's where the balance lies. And in thinking about therapeutic effects, we have to be mindful of that balance and probably arrange for drug delivery that's consistent with it.
Thank you. I think we have lots of active participants from various sectors, right? Like from the universities and recently from the Ministry of Public Health.
That's
So this is a very, great opportunity.
Yes, please.
Yes, please.
Please, sir.
Professor Bruce Beutler. So happy to listen to your lecture. It's not only great honor, but also very exciting to learn about your discovery. As a medical doctor, 50 years ago, when I started my career as a medical doctor, we had faced with what we call infection and malnutrition. And we talk about the relationship between malnutrition and infection. But today, you give us what we call enlighten us that infection and inflammation is different story. But it has some more or less some kind of relationship also. So that I try to understand your good lecture, and then as a public health man, I'm trying to think about this kind of discovery that it will be very beneficial for the public health in the future, or even in today, because in many developing countries or underdeveloped countries, there are still many infectious diseases. But as I mentioned that 40 years, 50 years ago, we are working in Thailand in rural area. We faced with the infectious disease. You mentioned typhoid, cholera, diphtheria, or many kind of infectious disease. As a young doctor at that time, the prescriptions always antibiotics. But now I realize that inflammation and infection is different. So if I had the knowledge at that time, we would save a lot of money of the patient. So I appreciate your lecture. Congratulations also. Thank you. Thank you very much for your good lecture. Thank you.
Thank you very much. Thank you.
Thank you. What about people from this corner and that corner of the room? So please feel free to raise any question. We also have our staff to deliver microphone to you. Yeah.
Good afternoon, everyone. My name is Miss Kanita Rungchamkan, and I am a master degree student from Faculty of Pharmaceutical Science, Naresuan University. And actually, I am interested in infectious disease, especially about pandemic. So here's my questions. According to the smallpox epidemic or the Spanish flu that used to happen in the last century, which caused a huge mortality in population, do you think would it be possible to occur again in the near future, and how can we prepare for it? Thank you.
Well, thank you. Yes, I do think that there can be great pandemics again. We witnessed ourselves, well, maybe you didn't. You're young enough that you didn't see the start of the HIV epidemic. And there was a time early in the epidemic when no one had any inkling about what caused that disease. Fortunately, that was the 20th century. There were good molecular biology tools, and the virus was isolated in a fairly short period of time, and amazingly, pharma was up to the task of making drugs that would at least control the infection, even as monotherapy, for a while. It's just remarkable how now we have highly active antiretroviral therapy that's really effective against HIV. Not that it isn't a problem. It certainly is still a huge problem, but it's not like what it would have been if it had happened in the 18th or 19th century. That's for sure. Pandemic flu could happen again, like 1918, could even happen worse than 1918, for all we know. And that's a matter of great concern to everyone. It's something that would obviously spread much faster. It could cause huge death, and certainly we should try to be prepared for it as best we can. Now, in a way, we do prepare for it by such devices as predicting the epitope specificity of this year's seasonal flu, But we could be surprised. It happens quite regularly. We could be caught unprepared, but we should at least have the capacity to make antibody quickly, to make protective antigen quickly if the need arises. Also, there's a great way to go in making pharmacologic therapy for infectious flu. We have Tamiflu, we have some other agents, but there's nothing that says we couldn't do a lot better than that. And those are the two ways I can think of to prepare. But I must say, everyone should not discount the possibility that there could be a catastrophe from that.
Good afternoon. I'm Dr. Prateep Wanisorn from Department of Medicine, Naresuan University. As you have been exposed to research since a young age, and I would like to ask what or who inspired you to become a very accomplished scientist today?
A big contributor was my father, beyond a doubt. He was a biomedical scientist. He was a hematologist who was quite eclectic in what he worked on. Iron deficiency anemia, red cell enzyme deficiencies, some leukemias, and glycolipid storage diseases. And one would ordinarily think such a person a dilettante, but he really went into considerable depth in all of those areas and was a renowned hematologist. From the time I was quite young, I remember having conversations with him about evolution, about various issues in medicine, about areas of science that neither of us knew very much about as well. But it was inspiring to talk to him, and certainly he must have influenced me a great deal. I had great teachers as well, a great genetics teacher, Dan Lindsley, who taught me the value of Drosophila and the forward genetic approach, or at least he tried to. I'm not sure I absorbed it all from him when I was in college, but being exposed sometimes makes a big difference, even if you don't go to work in that way right away. I had other teachers in medical school who were valuable to me also. Patricia Spear, Susumu Ono, many others. And so it's teachers and my father that certainly influenced me. None of us grows up in a vacuum, and everybody needs some kind of role model to look to. Usually a living person, but not always even that.
Yes, please.
My name is Somkiat Setpakdi, formerly from the University of Pennsylvania in Philadelphia and also Chulalongkorn Hospital. I'm intrigued by the last questions about, I would like to ask you, when you were a neurology fellow, resident, why did you change course to take up fundamental research in molecular biology? Because neurology many years ago was regarded as the prince of medical specialty. In fact, Sir Macfarlane Burnet, I was lucky to be one of his last students before he retired. He wanted to go into neurology, but as he was not the top student, he could not get the post. So he went into chemical pathology and did research in influenza and later on immunology. So I'm interested, what made you not pursue further in neurology and went into immunology?
It's a good question. I'll give you the honest answer. I went into neurology probably for all the good reasons you can imagine. I was fascinated by what I read about strokes that caused alteration of higher cortical function. I wanted to know how we think, basically. I wanted to know about that great mystery of consciousness. And I went that direction thinking that perhaps there would be technologies one day that would let us answer those questions. But clinical neurology was a bit disappointing for me in certain ways. I found that those syndromes were not quite as pure as they were represented in the textbooks. I found that for the care of strokes, epilepsy, all of the neurological problems I had to treat, there was a rote solution, and I found myself following A, B, C, D to diagnosis. It wasn't so much that one couldn't treat all of those diseases, because there was therapy, at least in the case of epilepsy. It was quite satisfying to see the result. But I didn't see an opportunity to move much beyond that and be creative and do experimentation. And that's what I missed. And I decided that would be a good stopping point for me, to find a fellowship in a lab once again, and possibly working with immunology once again, as I'd had some exposure to that before. So that's why I drew the line after 1 year of neurology, and I moved on.
Yes, we have a question at the middle aisle, please.
Hi, my name is Chairat Uthai Pibut. I'm working at National Center for Genetic Engineering and Biotechnology in Thailand. In the lab, we are working on drug discovery for malaria, which is one of the big infectious diseases. And actually, with last year at the American Society of Tropical Medicine and Hygiene, Bill Gates said that he'd like to see malaria eradication, within his lifetime. I think with all the technologies we have at the moment, what do you think that we can eradicate any big 3 or big 4 infectious diseases in the world, or, well, in my lifetime, in another 50 years? Would you think it is possible to, see something big news that we can get rid of?
I'm not prepared to say it's impossible. That's always a foolish thing to do. But I think it would be very tough, and let's say I can't see right now how it is possible with malaria, given that you do have animal reservoirs, given just the problem in devising a vaccine for malaria, which seems to have very good mechanisms to beat the adaptive immune system. To make a really fully efficacious vaccine has always been out of reach. But I'm not in that field, not directly, and you may know something I don't, so I won't be too dogmatic about it.
Any more questions?
We really have,
Yes, sir.
the active participants. Okay.
My name is Sid Pakdi. I'd like to ask you a serious question.
That was a serious one.
Professor Kase mentioned that the nutrition may have a part to play in the host response to inflammations and infections. So I'd like to ask you, what are the factors, important factors, you consider that may substantially influence the response of the host to microbial entries into the body or its toxins?
Well, I think the question might be posed in reverse as well. We know of a few things like zinc that seems to be important for some innate immune functions. Of course, iron may have a counterproductive effect with some infections. If one has too much iron, it can be bad. But we don't know a whole lot about it, I have to say, about trace nutritional factors that are influential. What we do know, though, I suppose, is that someone has a marginal nutritional status. They're not going to do as well surviving an infection. I think that's fairly clear. And that somewhat confounds the picture if we're thinking that it makes them more susceptible, and yet we know all along that it makes them less likely to survive a serious infection. Then we have two things going on. You may have some reply to that, and I'm happy to hear.
Thank you very much for that serious question. Any further serious questions? And may I say that this will be the last question, so please don't miss your chance if you would love to, raise your question or share your opinions.
Ponchuli Achawa Amrung, distinguished expert in comprehensive operations from Naresuan University. I wish to tell you that I am so fascinated by your eye-opening lecture this afternoon. My question, I would like to ask you as a layperson, because I'm not a scientist, I'm an educator. And if I got it right, I heard that you found something at the molecular level, something that it's called TLR, and it has signaling between particles of molecule at the molecular level, and that signaling helps sensing that something inflammatory has entered the body, and so the body can catch it and somehow get rid of it, or the scientists or the medical doctors can use it in their diagnosis and treatments of inflammatory infectious diseases. Am I correct?
You got it all right.
Thank you, sir. Well, I can be a scientist then, maybe next life. You also did mention that this sensing system can fortunately replace the use of antibiotics eventually, and that a lot of doctors are not quite aware of these new findings that you have discovered for the world. I am very impressed that you, as just one human person, can make such a big difference in saving lives of countless sick people all over the world. And so my question is that, how can you, I know that scientists are very focused upon the research, but if you want to make a big difference in practicality, how would you go about it to educate doctors all over the world to come round and have a shift of paradigm, come back and use what you found in practice and stop using antibiotics that we know that are not very good to us? Thank you.
I didn't say that it could replace antibiotics, this system, so there may be a little misunderstanding there. The first part of what you said was all correct, but I don't view this as a way to replace antibiotics, not at all. It may be that we need to make new antibiotics to replace the ones we have that lose efficacy. That may be the source of confusion. In general, there is a divide between basic scientists like myself and medical doctors, and that's growing wider and wider. And it is a very legitimate point you raise. The NIH tries to address this by training clinician scientists, and yet that doesn't seem to be necessarily the answer either. And I don't say that I have the answer, but somehow there does have to be transfer of information from basic scientists to both physicians and to pharma. And it gets to be more and more difficult all the time.
Thank you very much. Thank you very much for all the questions. I think we have, serious and impressive questions, and we really have active, participants. And definitely, we really have a good time attending this very special lecture. And now we have reached the end of the event.
Yes, Ms. Pong, I feel very fortunate indeed to witness and to join this event and have a chance to listen to Professor Beutler. Yes.
And I would love to also thank you, the main sponsor once again, Cognita Teaching Experience, Centura Hotels and Resorts, and Millennium Hilton International Peace Foundation, Burapha University, Mahasarakham University, Naresuan University, Srinakarin Virod University, Thaksin University, and all people concerned. Thank you for your support and making this event happen.
Okay, a big round of applause, I think.
Exactly.
So, May I please ask all the participants to fill in the evaluation forms before you go, and you can also scan the QR code or follow the link just in front of your event folder.
And now, I, Ms. Pongam Liamsirit Watana and Dr. Chayanin Pratumsood, we have to say goodbye to you.
Thank you very much and goodbye, and have a safe journey home. Thank you.
Goodbye and see you next time in the 6th ASEAN series. สวัสดีค่ะ